totally change? I mean, is there any kind of a product made in CHO cells that you couldn't make with this technology?
No. So anything you can make in CHO cell, you can make in here, because the CHO cells are making antibodies.
Okay.
And once you get an antibody sequence, we can make full-length ones. We can make single-chain antibodies. We've shown we can make all of those.
Okay. So now that you would have to transfer that to the manufacturing stage now that you think you've got the technology to where you've proven it, you've actually shown in mice that you can get the therapeutic effect. And now to get this into manufacturing, to actual technology, what do you have to do?
Probably go get a new target. Okay. Because drug companies will never reinvent the same drug again. They've already sunk huge amounts of cost into making an anti-CD22 with a toxin on, so they're not going to make another one that's a little better. Okay?
Yeah.
So what we're going to have to do now is sit down with oncologists and say, so the reason we made this one, rather than go make a new one, because there's a lot of cancers that are untreated, either because they're too small a patient numbers that the big drug companies haven't gone after them, or because they're just difficult to make a protein against. And we thought about, maybe we should go make one of those, a brand new, and show that we can make a drug nobody else made. But the problem with that is if you go make one of those, even if it works, then the pharma industry kind of says, well, how do we know that this doesn't work just because you picked that target? So the reason we picked CD22 is because it was in clinical trials. So we wanted to do a head-to-head, apples-to-apples comparison. So we could say, look, here's in the literature how these drugs in clinical trials work. Here's how ours work. Now the pharma industry, NIH, they can look at this and go, okay, yours works just as good, right?
And then they can say, why not manufacture this way?
Yeah, so, but when they manufacture it, it won't be, let's make a duplicate of one that's already out there. It's like, okay, we see that your drug works just as good. We see that you can manufacture much cheaper. Now let's go and look at all the other ones that we might make. So that'll be next generation. That'll be what we do. And personally, for me, I want to make dog anti-cancer antibodies.
Because?
So there is nothing that you have to do in a human, right, if you want to make a human drug, that you don't have to go through a dog model anyway. Dogs have the exact same cancers we have. But no one will make an anti-cancer antibody for a dog because no one would pay $10,000 or $100,000. But if we could make one for a dog, and we really can make it as cheap as we think we can, now instead of being $50,000 or $100,000, we think we can get this down to a few $1,000s and maybe even cheaper. Now you have something which dogs would buy for their humans would buy for their pets, right? The dogs won't buy.
That would be within a cost that a lot of people just might decide to pay.
Exactly, right? I have an old Bernese Mountain Dog, right?
Okay.
So I have her in at the vet a couple weeks ago because she's developed she has now a high squeaky voice, and it turns out when she barks it's really high, and it turns out it's something that Bernese Mountain Dogs get when they get old. So it's nothing to worry about, and there's no treatment for it. It's just like, no, it's called pharyngeal paralysis. Their pharynx starts to just get paralyzed as it gets old. So it makes so she barks like a little dog. It's kind of funny. She's an enormous dog with a high. But anyway, I had her in there, and I'm waiting in the waiting room just to talk to the MD, and there's a guy sitting next to me with this beautiful Labrador, right? Nice little female lab, about 5 years old. And he says, yeah, unfortunately, she's got cancer, and I just can't afford the treatment. You can get chemotherapy for dogs.
Yes.
But it's about $6,000, and all it does is slow the tumors down. It doesn't really cure them. Yeah. And I'm thinking, you know what? This is a cure. This isn't, give a chemotherapy and slow down the tumor. We have a chance to really cure these things.
So just in the process that you would need to take a drug to humans, you can already do the work for canine therapy.
Exactly. You have to go through canines anyway to prove they're safe and effective and everything else, right? So I just think you would never do this with traditional technologies, right? Yeah. Because you could make the best canine drug in the world, but it's too expensive. You'll never sell it to them. But what we could do is we could make one for canine lymphomas, actually make it cheap enough to sell it, and then that is the proof of concept that we should make these things for human. And now, more important than that though, not only have we proven that the system works, we've proven we can do it for dirt cheap, right? Because people will never let you do it for dirt cheap in humans.
Yeah.
Because that's not what FDA does. We have to have no risk. We don't care if it costs $600 million to do this study. You can't make one person sick.
But if you say you might be able to give this treatment, this cure for certain cancers, and do it for a couple thousand.
Yeah.
And then people will say, why can't I have that?
Exactly. That's exactly what I want to do. What I want people to say is, wait a minute, how come we can do this in our dogs for $2,000 and it costs $100,000 in humans? And then I will say, yeah, why does it? Let's ask these guys. So that's my goal. I'm trying to undermine all of medicine in this country.
That's a hugely ambitious goal. But by doing it through animals, which you've got to do anyway.
Yeah.
Yeah. So have you made any commercial contacts on this yet, say for any canine therapeutics?
We sent emails out to about 4 or 5 different vets who we saw were treating canine cancers, and they all said the same thing back. They said, oh, all of these therapies work, they're just too expensive for dogs, we can't do them. And I would write back and I'd say, no, that's the point. My technology is so cheap we can't, and none of them got it.
Oh.
There was one guy at Davis who I think kind of got it. He was like, oh, yeah, this sounds really exciting. Could you really make them cheaper? I'm like, yes, we can really make these cheaper. Let's do this. And he sort of agreed to do it, but we have what's called tech transfer offices at universities.
Yes, and so kind of bogged down.
Yeah, their tech transfer gal didn't get it at all.
Oh.
She's like, well, we have monoclonal antibodies already. What's your technology? And I tried to explain it to her, but she just didn't get it.
That's kind of disturbing in a tech transfer person.
Oh,
yeah.
Tech transfer persons at universities are generally the worst. They just don't get. I mean, we just had to completely revamp ours here at UCSD because it got to be so bad we weren't licensing anything.
Oh, that's not good.
Yeah, they all have this kind of attitude that. I think what it kind of is
Do
no
you
one
mind
ever
saying
lost...
that on the record, by the way? Because I might want to put some of this online.
Yeah, let me say it as generic, okay, rather than UCSD.
Oh, okay,
right?
but you've got...
generically, the biggest problem with university tech transfer offices, right, is they have no ability to really judge my science. Because it's very hard to do, right? The stuff we work on is super sophisticated, and these people aren't scientists. How can they know the value of it? So what they think is, well, what if I license this for really cheap and it turns out to be the greatest product ever?
Yeah.
And then everyone will come back and yell at me and I'll lose my job because I gave away all this great technology, right? So instead what they try to do is they try to license it for really high rates.
And then you don't...
And then no one gets it because people are like, it's experimental. I'm not going to pay you a lot for something that doesn't work. So because of that, nothing was getting licensed. So we completely revamped ours this year. And Jane Moores, who's our head of tech transfer, and a bunch of us scientists got together, and we told her, Jane, the goal shouldn't be to make money for the university. The goal should be to get our technology out into the commercial sector so it benefits society. So stop trying to think about how you're going to make money for the university and start trying to think, how are we going to deploy this technology as fast as we can? It's an enormous win for the taxpayers if my technology gets out and is utilized and saves people's lives, even if the university doesn't make a penny. Because if it doesn't get out, then I guarantee you we're not going to make a penny and no one's life is going to be saved.
And isn't it possible, too, that if you give a low license fee, it would be used a lot?
A lot more, exactly. And so a couple of universities do this. Stanford and MIT are the two famous ones.
I think there was an example, actually, that I wrote about some years ago where it was some technology that just expired maybe about 10 or 11 years ago, that it was a basic biotech technique.
Yeah.
It may be related to making recombinant organisms, and it brought in like $200 million because they licensed it very cheap and everybody wanted to use it.
UCSF, yeah.
Okay. So... can you kind of walk me through now the technology about